Sorry, my co-chair doesn't seem to be here, so I'll be... Oh, she's coming. I'll keep going on. Very happy to have you today here in Paris and to open this session on the on a new era in reproductive genetic testing. And we'll start with a presentation from Sheila Noak, survivor from Canada, survival of the fittest, unraveling aneuploid cells depletion in mosaic embryos. Sheila, the floor is yours.
Thank you for your introduction. Hi everyone, my name is Sheila, and I'm a PhD student from the Create Fertility Center in Toronto, Canada. And I would like to thank Eshre for this opportunity to share my work today. So aneuploidy is a hallmark in early human embryo development. It can lead to aneuploid embryos or mosaic embryos where euploid and aneuploid cells can coexist. The incidence of aneuploidy is clinically linked to adverse pregnancy outcomes, and it becomes more frequent as women age. And we can see from this trend here, due to a higher error rate in oocyte development. On the other hand, mosaicism is not really related to maternal age, and we can see from this yellow line. So for patients without euploid embryos available, mosaic embryos are used for transfers based on recently established clinical guidelines. So how did these mosaic embryos perform? And here we can see the transfer outcomes of more than 4,000 mosaic embryos documented in the international registry up to this year. So first we can see mosaic embryos in general have a 40% clinical pregnancy rate comparing to 50% in the euploid embryos. And interestingly, only 1.2% mosaicism persisted in the pregnancy. Babies born from mosaic embryo transfers also show no significant differences in terms of birth weight or gestational length comparing to the euploid embryos. So this indicates mosaic embryos have the potential to result in completely healthy babies, even though they have a slightly lower clinical pregnancy rate. And all these clinical evidence pointed to a potential self-correction program in these mosaic embryos to deplete the aneuploid cells, but the mechanisms are still unknown. So what happened to those aneuploid cells, and what is triggering aneuploid cell depletion in these mosaic embryos and allow for pregnancy success? We believe it's very important to understand these clinical outcomes also from a molecular perspective. So in this study, our goal was to investigate the ploidy-specific transcriptomic signatures in post-implantation human embryos. This will allow us to identify shared molecular mechanisms that could be driving aneuploid cell depletion. And finally, we hope to validate these findings using in vitro models. So first, we culture human embryos from the pre-implantation stage to the early post-implantation stage by adapting the protocols established by Shabazi and Xiang's team. We also performed immunofluorescence staining on a J10 embryo that was cultured in our system, where we validated the presence of 3 essential lineages: the epiblast, the hypoblast, as well as the trophoblast. After the culture, we performed single-cell RNA sequencing on the embryos, and then we demultiplexed the data to identify the embryo regions, and also infer CMV to identify the aneuploid cells. So from the single-cell RNA-seq data, we can first see that we identify gene expression profiles specific to 5 major lineages that correspond to the early post-implantation stage. This includes the epiblast, the hypoblast, as well as 3 different trophoblast subgroups: the cytotrophoblast, extravillous trophoblast, as well as the syncytial trophoblast. So now with this gene expression information, we can infer the copy number variations in the single cells using an established pipeline called infer CMV. So we basically chose cells from the euploid embryos as the reference, and then identify the cells with abnormal copies of chromosomes based on the CMV proportion on each chromosome. And from these aneuploid cells, a total of 14 chromosomes are involved with the dominance of chromosomal gains. And then we classify the embryos based on percentage of aneuploidy. So in total, our data set included 7 aneuploid embryos, 16 euploid embryos, and 8 mosaic embryos. So all 3 types of embryos had the ability to generate all those lineages that we previously identified, as you can see from this U-map here. But at the same time, we're curious to see how aneuploidy can affect the further downstream progression of these lineages. So we performed pseudotime analysis on the cells from the epiblast, the hypoblast, as well as the trophoblast lineages. And then we found that there was a significantly higher proportion of cells in the aneuploid embryos, which is indicated by the red color in each of these plots here, remained at a less differentiated state comparing to euploid or mosaic embryos. So this indicates even though aneuploid embryos had the ability to generate those different lineages, they can still experience developmental delay due to impairment caused by aneuploidy. So this finding led us to investigate how different embryo environments can potentially affect aneuploid cell fate, because the aneuploid cells seem to behave differently when they're in an aneuploid embryo versus mosaic embryo. So first, we performed differential expression analysis between the aneuploid and euploid embryos to see how aneuploid cells will respond to a homogeneous environment. At the same time, we also compare between the aneuploid and euploid cells of the mosaic embryos to see how aneuploid cells will behave in the environment that euploid cells also coexist. And then we narrowed down to a couple interesting markers that were differentially enriched between these 2 comparison groups. So some of them are related to adaptive stress response, such as unfolded protein response and regulation of protein processing. And these are all essential functions that are important to maintain cell survival or fidelity, especially under aneuploidy stress. And we can see that they're all upregulated in the aneuploid cells of the aneuploid embryos, indicated by the lighter gray color, but suppressed in the aneuploid cells of the mosaic embryos. At the same time, we also found markers related to pluripotency regulation. So for example, the functions important to maintain the pluripotency states are upregulated in the aneuploid embryos. But the functions that are related to cell differentiation or lineage specification, they're upregulated in the aneuploid cells of the mosaic embryos instead. So this suggests the aneuploid cells could be driven towards pluripotency exits when they are in the mosaic environment. So these findings show that when aneuploid cells are in a homogeneous environment, there could be temporary adaptive mechanisms that help them tolerate the aneuploidy. But if they are in the mosaic environment, they can experience functional suppression that leads to their diminished survival advantage. So this kind of differential fitness levels in the mosaic embryo can contribute to cell competition between the euploid and aneuploid cell population and eventually induce the clearance of aneuploid cells, which is likely the less fit population in this case. So now we want to validate these findings from the embryo data, and then we wonder if we can recapitulate this mosaic embryo environment using an in vitro model. So we set up a transwell system by co-culturing euploid and aneuploid iPSCs to mimic the mosaic embryo environment, given the functional similarity of these cells to embryonic cells. And correspondingly, we set up control transwell system to mimic the aneuploid and euploid embryo environments. So in this experiment, we chose trisomy 21 first as the aneuploid cell population. And in this first plot here, we can see the growth rates of the aneuploid cells between 2 different conditions and across 96 hours of co-culture. And then we can see the growth rate started to diverge after 48 hours and appear more robust in the homogeneous condition, indicated by the gray color that mimic the aneuploid embryo environment. In contrast, if we look at the euploid cells, the growth rates are very similar between the two different conditions. And then it seems that the mosaic environment is having a larger impact on the aneuploid cells in this case. And finally, we looked at the expression of a stress adaptation marker, DDID4, autophagy marker, LC3, as well as the apoptosis marker, caspase-8. And here we can see their expression intensity in the aneuploid cells of two different conditions. On the top panel, we have the homogeneous condition, and at the bottom, we have the mosaic condition. So after we quantify the intensity, here we can see a log2 fold change of these fluorescent intensity in the aneuploid cells of the mosaic condition. So we can see downregulation of DDID4, LC3, and upregulation of caspase-8. If we compare to their euploid neighbors in the same mosaic condition, we can see DDID4 is significantly lower in the aneuploid cells. Caspase-8 is also significantly higher. And this shows that there could be suppressed stress recovery mechanism and increased apoptotic signaling in the aneuploid cells when they're in the mosaic environment. And finally, we looked at the expression of two pluripotency markers, OCT4 and CLIF4. So CLIF4 is downregulated in the aneuploid cells of the mosaic condition, and also compared to their euploid counterparts in the mosaic condition, it's also significantly lower than the euploid cells, whereas OCT4 stay pretty consistent throughout different conditions, and this is a general pluripotency marker. And this kind of suppressed naive expression that we see here align with the mosaic embryo finding, where we see the aneuploid cells seem to be driven towards differentiation when they're in the mosaic environment. So this indicates mosaic environment can potentially attenuate aneuploid tolerance, leading to aneuploid cell depletion. So in conclusion, our study here showed different embryonic environments can contribute to distinct transcriptomic patterns and therefore survival outcomes of the aneuploid cells. And we set the groundwork to explain how mosaic embryos can self-correct and develop normally. And finally, we can potentially inform on the range of viable embryos in future clinical practice and also improve fertility preservation options. With that, I would like to acknowledge my PI, Dr. Clifford Liebrecht, as well as my mentors, Drs. Valentin Marcinkova and Dr. Stuart Russell, as well as everyone from the CREATE and the Zuvis team. Thank you for your attention. I'm happy to take any questions.
Thank you, Sheila. We have time for one or two questions. Is there a question there? We can't hear you. Can you?
How far do you think that our culture condition in IVF will improve aneuploidy in embryos?
The culture condition in IVF?
Yes.
The co-culture.
Do you think that our culture condition in IVF will increase the aneuploidy rate in embryos, or will be not in favor of survival of haploid cells?
So I think the main cause of aneuploidy is still dependent on oocyte quality and also maternal age. So I think culture condition might have minimal effect on aneuploidy induction. So I think if we control for other risk factors such as age or maternal drug use, we can potentially mitigate those risks.
But do you think that, yes, this I know, which are the original cause of aneuploidy in the embryo, but do you think that the culture condition also will help to survive or not survive, or a self-correction of this mosaic embryo in vitro conditions?
So you mean the standard culture?
Yes.
Yeah, so I think after implantation stage, self-correction can potentially happen during that window because the aneuploid cells are being depleted. And as we can see from our embryo that was cultured in our system, a lot of the mosaic embryos actually resulted in euploid embryos. within the 14-day window after we call the CMV using RNA data. So I think the extended culture can also help them to deplete the aneuploid cells over time.
But after implantation, you don't think that in vitro until the blastocyst stage, the culture condition would favor or not the self-correction of the embryo?
I think you mean the pre-implantation stage?
Yes.
It can also happen, but because PGTA was performed before the transfer, and it provides like a snapshot of what the ploidy status is like before implantation. So at the post-implantation stage, I think based on the CMV calling and expression data, it's more representative of the entire ploidy status. So I believe aneuploid cell depletion can happen within that window.
Thank you.
Okay, one quick question and one quick answer. We already late.
Stéphane Diville from France, Strasbourg. I have, here I am. Hello, here I am.
Over there.
I have a semantical question to ask you. Do you think self-correction is a proper term to use for this phenomenon you observe? Because for me, self-correction would mean that there is an active mechanism dealing with this problem and eliminating the cells. Don't you think it's just a dilution of the cells and it's a passive mechanism and not an active mechanism? So I really wonder that I do think that self-correction is not the proper term to be used in this situation. Can you comment on that?
So you mean self-correction is not a... sorry, do you mind repeat your question?
I think we should not use, on what you exposed to us today, you present, we should not use the term self-correction. It's not a self-correction. It is a correction, I agree with that, but it's not an active mechanism. For me, it's not, no one has never shown that it is an active mechanism.
The stem cell model, you mean?
Sorry?
That I use, the iPSC model? Or?
No, in the embryo, the embryo don't self-correct. It is corrected, but it's not self-correction. It's not
I see. You mean the term that I use?
Okay, I think we'll agree on that and we'll have to move on. Thank you.
We can talk about it later.
Yeah.
Thank you very much. And we will now move to the next speaker, Maria Berbel Fernandez from Juno Genetics, Spain. She will talk us about optimizing carrier screening panels, integrating 8,989 internal exomes, GNOME ID versus version 4 and advanced annotation tools for evidence-based peer-reviewed gene panel design.
Thank you for the introduction and good afternoon, everyone. Today, I'll walk you through our work on optimizing carrier screening panels, integrating our internal exomes, GNOME v4.1, and advanced annotations tools for evidence-based, peer-reviewed gene panel design. I'm a full-time employee of Juno Genetics. First of all, what is carrier screening and why is it so relevant? Well, carrier screening is used to identify individuals or couples that are at risk to have a child with an autosomal recessive or X-linked genetic disorder. The 2% of the couples are at risk of having a child affected with a recessive genetic condition. In developed countries, the 20% of pediatric hospitalizations and mortality are due to this type of conditions. And the 88% of children born with a genetic disease have no family history for the conditions. And as you can see in this chart, the prevalence of recessive disorders in affected births are much higher compared to the prevalence of chromosomal aneuploidies. And maybe you can be wondering, why did we need to rethink our carrier screening panels? And let me start with the current limitations, because there's a lack of a standardized framework for panel design, and because of this, there's very little consistency across the panels in the different competitors. In this study of Wang et al., they analyzed 22 panels, and they saw that only 15 genes, and that's just the 0.7% of the genes, were shared between all the panels. So these really make us question, are these panels truly evidence-based? Another important limitation is the use of outdated datasets, and also the inadequate ancestry representation. In this picture, you can see different NOMAD versions. NOMAD is a large genetic database that provides us with population frequencies. ACMG Tier 3 panel, proposed in 2021, was built in this NOMAD version, the version 2, that has a smaller size and a limited ancestry representation compared to the latest version, the version 4. So taking into account these limitations and that also we have now new resources like bioinformatic tools and resources for variant classification that didn't exist 10 years ago, with the large datasets that I mentioned, the version 4, in which we include a higher number of samples and also a better ancestry representation, all of these with our valuable internal data. With all of this, we saw the opportunity to improve our carrier panel design. And the 3 key objectives that we had are to enhance accuracy and inclusivity in carrier testing, to select genes based on a robust data-driven criteria, and finally, to optimize panel size while maximizing fetal disease risk. So to develop an evidence-based carrier screening panel, we wanted to maximize the clinical relevance and ensure the panel's utility in reproductive decision-making. So for this purpose, we used 3 gene sets. First of all, our internal panel, ACMG Tier 3 panel, and Mackenzie's mission. Mackenzie's mission is the Australian reproductive genetics carrier screening project in which they selected 1,300 genes associated with recessive and X-linked conditions. This gene list was curated by clinical geneticists, ensuring the clinical utility in reproductive decision-making. So in total, we started with a panel of 1,350 genes, because some of the genes are shared between all the panels, as you can see in this Venn diagram. So for our analysis, we used exome sequencing data of almost 9,000 subjects. And we included CNVs, and also we performed orthogonal tests to cover some technically challenging genes, as you can see here. Also, we used NOMAD data to have this panethnic approach in our panel and cover diverse ethnicities. We then extracted all the pathogenic and likely pathogenic variants in our target genes, the 1,350 genes. And for this, we used not only ClinVar, as it is common in other studies, but also we use an advanced annotation tool, that is Franklin classification. And for the conflicting variants, we developed a standardized framework in which 3 geneticists classified the variants based on ACMG guidelines. With all the pathogenic variants extracted, we then calculated the carrier rate by gene. Of course, we also reviewed the severity of the conditions included. And finally, with the fetal disease risk calculations, we obtained our proposed carrier screening panels. For the gene review process, which can be arduous due to its subjectivity, we use a standardized framework that categorizes diseases by phenotype severity. And this framework was proposed by Myriad Genetics. The review process included both an internal and also an external review. The external review was conducted by experienced clinical geneticists. And finally, we excluded 50 genes due to mild phenotypes and also for technical issues, like pseudogenes or variants that are not well covered by NGS. The fetal disease risk estimates the chance that a fetus will be affected by a genetic condition within the set of genes analyzed. And here, you can find how the fetal disease risk is calculated for autosomal recessive and X-linked conditions. As we are including both recessive and X-linked conditions, we use the cumulative fetal disease risk, that is simply the sum of the individual fetal disease risk by each gene in the panel. The cumulative fetal disease risk is represented in the y-axis in this graph, and the carrier rate, the gene carrier rate that I mentioned before, is represented here in the x-axis. And this is only an example, but it is important to highlight here that the biggest increase in identifying at-risk couples happens when adding conditions with higher carrier frequencies, from 1 in 100 to 1 in 200. And as we include rarer conditions to 1 in 200, from 1 in 200 to 1 in 1,000, we see that the number of individuals or the number of the couples identified increases much less. In other words, the incremental benefit of adding more genes drops significantly because the likelihood of both partners being carriers for the same very rare condition is extremely low. So in our analysis, we started with 1,350 genes, and we combined our internal data and also NOMAD data to have this panethnic approach. We then selected a cutoff of 1 in 500 gene carrier rate, and finally, we proceed with the fetal disease risk calculations. So the 90% of fetal disease risk includes 106 genes. The 97% of fetal disease risk includes almost 300 genes. And we can see that an increase of a 2% in the fetal disease risk would include more than 100 genes extra. We also wanted to compare our data with ACMG Tier 3 panel. This ACMG Tier 3, this panel, they recommended to include conditions with a carrier rate of 1 in 200. And this panel was proposed, as I mentioned before, in 2021, and was built on an outdated NOMAD version, the version 2. So they proposed 113 genes. However, when we analyzed our internal data and also the latest version of NOMAD, we saw that 11 genes, 11 of these ACMG Tier 3 genes, fell outside the threshold of 1 in 500 gene carrier rate. And we are not the only ones that are seeing this, because other studies, like this one of Smith et al., they identified 28 ACMG Tier 3 genes that fell below the threshold of 1 in 200 using the latest version of NOMAD. Same with this analysis, the Goldilocks approach, and as well as other studies like this one of Candoling et al. So to conclude, carrier screening panels should reflect current data and population diversity. Our approach increases both sensitivity and equity, improving the identification of at-risk couples across diverse populations. We propose a dynamic and a robust framework for carrier screening design, adaptable as new data emerge. And finally, carrier screening panels, including up to 300 genes, have been shown to be cost-effective in recent studies, supporting a broader implementation. Thank you, everyone, for your attention, and now I'll be happy to take any questions.
Thank you very much for the presentation. Are there already questions from the audience?
Sorry,
Please.
I don't want to be the only one to ask question. Considering the cost of the technology and the fact that you can have some specific population with a much higher risk for very rare disease, I'm thinking of area in the world where there is a tradition of consanguineous marriage. I don't see why we should limit ourselves in analyzing in the number of the genes that we want to analyze. And I know that, for example, for the Maastricht project, they use 2,500 genes, which cover most, if not all, the recessive and X-linked genes. So are you not afraid that you will miss some specific population with your approach?
Thank you. Yeah, thank you very much for your question. This is really interesting, and well, the whole point of the presentation is related to this question because, as you said, there are some populations in which the consanguinity is really common, but this panel is more for the population in which we consider that there's not such grade of consanguinity. And in this case, our study, what's proposed is to include genes with higher carrier frequencies because, as I mentioned, the likelihood that both partners are being carriers for the same condition is extremely rare. And the balance between we don't want to have an expensive carrier screening panel because we want to offer this panel to the majority of the population. So for the regular population in which the consanguinity is not as common as in other countries, we think that our approach is really valuable because we are covering the majority or the high frequencies, the conditions that have high frequencies compared to a larger panel that can cause a variant bottleneck and also a higher cost in variant interpretation. And also it's important to highlight here that when we are talking about genes that are less known, the variant interpretation is really complicated, and even more in the carrier screening context that we are only reporting pathogenic and likely pathogenic variants. So as the rarer is the gene, the less knowledge we have in the literature, and also the more difficult is to report a pathogenic variant in that gene. So even if we include more genes and also genes with, 3,000 genes in our panel, but we don't have evidence and we cannot report pathogenic variants in those genes, we will have exactly the same problem.
Thank you. If there is another question, it needs to be a very fast one. If not,
okay.
Thank you very much.
Moving on to the next speaker, I'm very happy to welcome Kaylin Reddy from Juniper Genomics, USA, and she will give a presentation entitled Whole Transcriptome and Genome Sequencing of Standard Embryo Biopsies Can Potentially Reduce Failed Euploid Transfers.
Yes, thank you, and thank you to the team here at Eshre who accepted this for oral presentation, and of course my team that helped work on this study. So, first, I am an employee and stock option holder at Juniper, which developed this test. More importantly, we're going to back up before I get to all the really exciting stuff about whole transcriptome and whole genome sequencing. We're going to start from the beginning, which is with PGTA. And I really want to emphasize, right, we can do really cool things these days with genetic sequencing, but we have to keep in mind why are we doing it. And I'm trained as a genetic counselor, so this is especially important to me. I like to think about why are we using the technology, how are we making patients' lives better. So in the case of PGTA, it was initially developed to assist with prioritizing embryos for transfer, and it worked. The live birth rate per transfer over time increased from about 30% without PGTA and up to about 55% after it was added. Okay, now we're going to do a little audience participation. Show of hands, and it's based on SART data, which is in the US, is the current live birth rate, and current because the data is kind of old, is from 2023. Per transfer, lower or higher? Who says lower than 55%? Who says higher than 55%? All right, mostly I think people are saying lower. It's a bit of a mix. It's a little lower. So PGTA was great. It worked until it didn't, and it really does still work. It's better than doing nothing, but the live birth rate has really stagnated. It has not meaningfully changed since about 2016. And so you can imagine as a patient, you are undergoing this complex process that is IVF, you're doing genetic testing of your embryos, and the best we can do for you is about a coin flip. So we have to ask ourselves, like, what is going on here? Are there persistent maternal factors, or is there something else happening? There was a study recently that demonstrated that failed transfers are rarely related to maternal factors. In fact, if you have enough euploid embryos and you keep transferring them, about 95% of patients will eventually achieve a pregnancy. But keep in mind, that is 5 euploid embryos. That means you had 5, which some patients don't. And that means you had to potentially go through every single one of those transfers. And we know that that is hard on patients, and not only from an emotional perspective, but also potentially from a financial perspective. So if it's not mom, what could be going on? We think that it could be genetics. And so the way I often explain this to patients, again, genetic counselor here, if we think about PGTA, what we're doing is we're counting books on a bookshelf. We're looking, are there 46 books on the bookshelf? But what we're not doing is taking all of those books off of the bookshelf, opening them, and reading them from cover to cover. That's what we're trying to do with whole genome sequencing, a lot more data. So then the next question is like, great, we can get the data, but does it matter? So there are a number of studies like this. I pulled this one. This is showing 200 patients who underwent miscarriage and donated the sample for genetic testing. These happen to be trios, so they tested mom and dad and the tissue. And they first ruled out any aneuploidies. And then of the remaining, we see that there are about half of them that can be explained by either a likely pathogenic variant in a known disease gene or a candidate variant in a known or novel gene. So we know there's something else going on here in these losses beyond just chromosome abnormalities. So based on all of that background, we decided to move forward with our study. We had 30 donated embryos from 7 families. These are donated embryos. All of these families were finished having their children and had remaining embryos. That means many of them were aneuploid, but we did receive also some euploid embryos. For all of the embryos, we had at least 2 samples per embryo, so at least 1 biopsy and the remainder, and in many cases more than that. For all of the embryos, we performed whole genome sequencing and whole transcriptome sequencing to a 50x depth. And then, again, trios. So we also performed trios in this study. We need parental blood samples. That was important to us because it allowed us to look for de novo variants in the embryos. This is my favorite part because I've done variant curation in my past and really thought about even carrier screening panels like the last speaker was discussing. So the first question is like, well, what genes really matter? How do we even know what we're looking for? What will we report on? And there have been a number of studies and progress in this area in 2 ways. First would be the study that I mentioned earlier, and a number of other studies like that are performed on products of conception and identify variants. And so we can look to those genes. It's kind of commonly called the intolerome. You can Google it and see a whole list of these things. The other place we looked is there are a number of online databases and publications based on CRISPR studies that tell us what genes are doing what inside the body. So now we know potentially what genes matter. Now we have to figure out if there's a variant in that gene, what variants matter. This is a little trickier. For anybody who's done variant curation, you know that some of these variants are really obvious, like half the gene is missing, and it's easy to be like, that is probably a problem. And many of the variants are missense or small indels. And so in those cases, we're looking at computational evidence, and we're also looking at a number of population databases. In the case of variant curation, though, for variants in embryos, we are actually not looking for the presence of those variants in databases. Rather, what we are looking for is the absence of those variants in those databases. Because what we're trying to find in these embryos are variants that might cause lack of implantation or a miscarriage or something like that. So by definition, we are expecting to not see them in the population. So here's what we found. Of our 12 embryos, 12 of 30 had variants, so about 40%. 7 of them had compound heterozygous SNVs, so a variant inherited from one parent, or from each parent. 6 male embryos had hemizygous X-linked recessive variants, and then 2 embryos had de novo SNVs. And again, we were able to determine that because we had the parental genomes. For those of you doing math out there, 7 plus 6 plus 2 is indeed 15 and not 12. And that is because a single embryo can have more than one of these things. So our conclusion based on this is that the frequency of variants is consistent with the solve rate in the product of conception studies. Remember earlier we discussed that of the 200 cases, about half of them were solved. It's also relatively consistent with the failure rate in euploid transfers. About half of them do not succeed. And so based on this, we are hypothesizing that prioritizing euploid embryos without the viability variants could increase the live birth rate and reduce our failed euploid transfers. More importantly, in bringing it back to patients, this is really aimed at trying to help them better prioritize embryos for transfer, get them the result that they actually turned up in clinic for, and save them time, expense, and anxiety. Obviously, there's a lot more that we could do here. We believe that we could have further reductions by including structural variants or CNVs. We really did focus, at least in this study, on SNVs and indels. And more importantly, what we need to figure out next is, now that we see these variants, when we actually go to transfer those embryos, what are the outcomes? Do any of the embryos with these variants actually succeed in a live birth, or do in fact they unfortunately fail? And those studies are currently underway. So here's the team at Juniper. Huge shout out to them. I joined this team late in October last year. They've been working on this for years. And also thank you to our patients who graciously donated their embryos and their blood so that we could perform this study. I'll take a question.
Congratulations, Karin, for respecting the time. That gives us some time for questions.
I promised you I would.
Maybe I'll start. Do you really think that the variants that you've identified could account for the fact that some of the embryo will arrest and will not implant and develop? Because the variants that are usually identified, they account for diseases that develop much later in life, and they're not expected to induce implantation failure or embryo development arrest.
Yeah, so we believe that some of these variants. To be fair, first we are taking these from blasts, and so they've at least made it that far. But potentially some of the variants, yes, may very well eventually result in arrest or the lack of implantation. And again, we're saying let's maybe try to transfer euploid embryos without the variants and improve the live birth rate.
Okay, one question.
Great talk.
Olga?
Olga Chuiko, yes. While I was walking, I forgot the question. What happens, or how would you deal if you detect a variant that is not present in the database, like you would expect, for example? But
all right, can you lower? The variant is not present where?
In the database, like you would expect, but also the gene is not known in the tolerome. How would you deal with those?
Yeah, I think in that case, we probably wouldn't report. We would wait until there's more information. If we can't say, I like to be able, as a genetic counselor, right, to think this all the way through to the end. So we need to be able to say something to patients about, hey, what does this gene maybe do? What do we think? Why do we think it matters? And then why does the variant matter?
Okay. No more questions?
Just shortly, here on your right. So do you actually think, or are you planning, or are you already doing, are you implementing this in the clinic? And would you recommend that RNA data are taken together into account for embryo transfer in short or long term?
Yeah, so We are working with a handful of clinics, and the patients I think that are most interested in this clinically are those that maybe have already failed a cycle, or we've all seen those patients that kind of come in wanting to know like literally everything they can possibly know. And so those are the patients primarily we've been working with. I believe you asked about RNA. Mm-hmm. So with respect to RNA, we are primarily using it to help us with the PGTA calls, almost like a second rebiopsy, if you will, for the PGTA. But my dream, because I am deep in variant curation, is to also be able to use it to help us predict the variant interpretations.
Good luck with that. Keep us updated.
Will do. Thank you.
Thank you.
Thank you.
And we move to the next speaker then. Oh, sorry, my overview. Oh, yeah. Nerea Castillo Barco, and she will talk to us about maternal plasma cell-free RNA as a liquid biopsy for first trimester screening of early and late onset preeclampsia.
Well, first of all, I would like to say thanks to the organizing committee for giving me the opportunity to present this research entitled Maternal Plasma Cell-Free RNA as a Liquid Biopsy for First Trimester Screening of Early and Late Onset Preeclampsia. But before starting, I had to declare that the results presented here are patented, and some of the authors, including myself, are employees of iPromom, the company that founded the clinical study. Having said that, let's start. Preeclampsia is a hypertensive disorder of pregnancy that arises after 20 weeks of gestation. And clinically, there are two major subtypes: early onset, diagnosed before 34 weeks, or late onset, diagnosed after 34 weeks. Traditionally, it has been attributed to placental dysfunction, but emerging evidence suggests or points out an upstream origin, points out to the maternal endometrium, the maternal soil, through decidualization resistance. Decidualization resistance refers to impaired endometrial cell differentiation during the menstrual cycle, which in case of pregnancy leads to abnormal presentation and compromises pregnancy health. Over the past decade, our research group has demonstrated the link between decidualization resistance and preeclampsia through a series of studies, including in vitro and animal models, as well as RNA in vivo endometrial sequencing. Earlier this year, we published a paper in Nature Medicine in which we deeply characterized this condition through multiomics, providing over 2,000 biomarkers. And our last research is being reviewed right now in Nature Communications, and here I will present the main result. Based on this body of evidence, we hypothesized that decidualization resistance during the late secretory phase disrupts the maternal fetal crosstalk from the early beginning of pregnancy. As a consequence, there is a unique RNA profile in the maternal bloodstream that can be detected and leveraged to develop a predictive tool for the early risk stratification of preeclampsia. To address this hypothesis, we conducted a prospective and longitudinal case control study across 14 tertiary hospitals in Spain. We collected peripheral blood samples from pregnant women at 3 time points: the first, second, and third trimester of pregnancy, or at the time of preeclampsia diagnosis. We recruited a total of 9,586 pregnant women to enroll at least 30 early onset preeclampsia patients. From this cohort, after excluding participants due to selection failure, loss to follow-up, or other pregnancy complications, we had a total of 7,142 eligible women. Among these participants, we selected a subset to perform the cell-free RNA analysis. And specifically, we included all early onset preeclampsia cases, which were 42, a randomly selected subset of late onset preeclampsia cases of 43, and 131 controls, also randomly selected and matched for key variables such as maternal age or BMI. The number of participants selected was established to achieve a ratio of 1 case to 3 controls, to have balanced groups. Regarding the clinical data, we had no significant differences for the gestational age at blood sample collection in the first and second trimester samples. We had significant differences at the third time point, but we don't use these samples to develop the predictive models and for further analysis related to the underlying biology, just samples that were matched were included to avoid bias. We had no differences for maternal age, parity, ethnicity, or BMI. Once participants were selected, blood samples were processed to isolate and sequence the cell-free RNA with two major goals. The first one was to map the cell-free RNA origin and dynamics. And to this purpose, we used the Human Protein Atlas database as a reference, focusing on those tissues that are directly involved in the pathophysiology of preeclampsia. Next, we performed differential abundance analysis comparing groups across pregnancy, including all the transcripts identified. The second major goal was to develop the predictive models, and to this purpose, we randomly split samples in two sets. The discovery set, used to select features and to train the models, and the validation set to test the performance in a holdout set of samples. Additionally, we perform an external validation of the predictive signatures using an independent dataset published by Moufarreg and colleagues at 2022. Now let's move to the results. As I mentioned, first we mapped the cell-free RNA origin using the Human Protein Atlas database. And this approach revealed that our experimental protocol captured over the 90% of transcripts identified for each tissue involved in preeclampsia. Next, we calculated a signature score using the normalized count. And if I zoom in this part of the plot, you can see how early onset preeclampsia cases had an increased signal from the liver, the kidney, and the decidua during the second trimester of pregnancy, indicating tissue injury up to 8 weeks before diagnosis. If we come back, and now we pay attention to the third time point, and I zoom in, you can see how at this time point, almost every organ or tissue analyzed had an increased signal in cases compared controls, and it was notable in early onset preeclampsia cases. Next, we performed differential abundance analysis across pregnancy, but here in the slide you can see the results at the time of diagnosis. Comparing cases against controls matched by the gestational age at blood sample collection, you can see how early onset preeclampsia patients had over 24,000 differentially abundant transcripts, whereas late onset preeclampsia exhibit approximately 12,000. Gene ontology analysis revealed the two subtypes had shared but also distinct pathways, highlighting that both subtypes had a signal of fetal damage, which was notable or higher in early onset preeclampsia, as well as this subtype had an exacerbated pro-inflammatory hallmark. Next, we focus on developing the predictive models. In the first trimester of pregnancy, we developed an early onset preeclampsia model comprised by 36 transcripts that showed a high performance around 18 weeks before the clinical onset. Specifically, we obtained an area under the curve of 88 in the internal validation and 87 in the external validation. For late onset preeclampsia model, we got poor performance, obtaining an area under the curve of 68 internally and 63 externally. During the second trimester of pregnancy, we were able to develop an early onset preeclampsia model comprised by 87 transcripts, which provide high performance stratifying patients around 8 weeks before the clinical onset, obtaining an area under the curve of 89 internally and 81 externally. For late onset preeclampsia model, we had a high performance model comprised by 92 transcripts with an area under the curve of 90 internally and 77 externally. To highlight that, early onset preeclampsia models had a high percentage of transcripts associated with decidalization resistance in the 2 trimesters, the first and the second one. This underscores the deciduous role in the pathophysiology of this subtype. In contrast, late onset preeclampsia model just includes 2 decidalization resistance markers. To conclude that a cell-RNA-based blood test in the first trimester can predict early onset preeclampsia up to 18 weeks before diagnosis, offering a valuable window for preventive care. The strongest predictive signals for early onset preeclampsia were linked to decidalization resistance, supporting a key role for uterine dysfunction in its pathogenesis. In the second trimester, cell-free RNA profiles distinguish early onset from late onset, highlighting distinct biological mechanisms behind each subtype. And to mention that currently we are conducting a large prospective trial with 7,473 participants to validate the test's clinical utility in real-world settings. Finally, I want to thank the wonderful team that has made this possible, including hospitals and clinicians involved. And thank you all for attending this presentation. And I'm happy to take here any question, but just in case we have not enough time, we will be at booth C27 after the presentation.
Thank you very much. And you were on time, so we can start with questions.
Hello, Alain Rico, Thermo Fisher Scientific. Very nice talk. Did you try on saliva instead of blood?
I can hear you well, sorry. Can you see it aloud?
I will ask again. Did you try on saliva instead of blood?
No, we tried just blood in our experimental design.
Okay.
We didn't collect that. We collect Buffy codes and plasma samples. But
and very quick second question, did you compare with biomarkers, the existing biomarkers for preeclampsia?
Yes, in fact, in our predictive models, for example, for the first trimester in early onset preeclampsia, we had some markers that has been linked before with the conditions such as MMP7 or PAEP.
Thank you.
Yes, please.
Yes, hello, thank you. Hanvik from Norway, very nice presentation. Did you do any genetic testing of the mothers or fathers? So do you have any DNA from them from before the cell-free RNA to see who could be at risk for developing the disease before they get pregnant?
Thank you for that point. We will have it into consideration because nowadays we didn't conduct that. To perform some associations analyses, we think we need even more number of cases. And with 30 early onset preeclampsias, where in this case we have 42, perhaps we couldn't find associations, but it's not because there doesn't exist, it will be because we don't have enough sample size to obtain something significant after the multiple p-value corrections.
Thank you very much. I also have a short question. You said you already started with a study to evaluate the clinical utility of this test. At which time point of the pregnancy, which week of the pregnancy are you planning to establish your sampling for patients?
In this case, we are doing the same design, so we are collecting again samples at 3 time points. First trimester, second and third trimester, and specifically in the first trimester we collect samples from 9 to 14 weeks because we try to adapt to the routine analysis that these women suffer when they go to the visits. So we are adapted to the protocols of the hospital, so when they went there we collect the samples. So that's the windows. 9 to 14, from 15 to 27, and after 28.
And are you planning to reduce this to 1 time point for clinical implementation, or is the idea to keep a follow-up throughout the pregnancy?
For us, basic research is extremely valuable, so we have different goals. And one goal is to develop a product for screening during the first trimester or second one, but we want to continue understanding what is happening in preeclampsia, so we consider valuable all these samples to analyze as much as we can.
Thank you very much.
And now we're moving to the last presentation of this session with advancing embryo selection, combined DNA and RNA analysis in PGT. And this is going to be presented by Bing Han from Spain.
Good afternoon, everyone. Thank you for the introduction. It's a true honor to be here at Eshre today. My name is Bing. Today, I'm very excited to share our work on the topics that's central to everything we do. advancing embryo selection. As we all know, PGTA has had a remarkable 30 years journey from sampling methods involving from polar body to T biopsy to detection technology advancing from FISH to NGS sequencing. It has improved our ability to screen for aneuploid. Despite all that progress, PGT still face some challenges. The first, we are working to really address those tricky false positive and false negative results that come from mosaicism. And second, our continuous goal is to improve outcomes by better selecting the most valuable embryos for transfer. The reason, of course, is that implantation is a complex process. It's a sophisticated dialogue between the embryo and the endometrium, driven by vast gene regulatory networks and metabolic pathways. This complex demands a more sophisticated approach. It requires us to look beyond just the DNA blueprint. This leads us to our central question. In addition to chromosome status, can we use the embryo's transcriptome, its active gene expression, as novel predictor of implantation potential? To answer this, we combined DNA and RNA sequencing in a single workflow. Let me briefly walk you through our study design. We analyzed 412 blastoses from the 102 patients. For each biopsy, we did something unique. We split the lysis into two parts. Part one, we used for standard PGTA to detect DNA copy number variations. And part two was used for RNA sequencing to explore the transcriptome. Our analysis had two main goals. The first, to characterize and compare the transcriptomes of different embryo types, euploid vs aneuploid, and day 5 vs day 6. And second, we use this data to build the predictive model that could identify the euploid embryos most likely to result in a clinical pregnancy. First, the baseline result. Our combined workflow was highly robust with successful CNV detection in over 99% of cases. Furthermore, chromosomes 16, 21, and 22 were the top 3 whole chromosomes aneuploids. And our RNA-seq data accurately reflected the DNA findings. We observed a clean gene dosage effect. Genes on missing chromosome segments were underexpressed, while genes on extra segments were overexpressed. This gives us high confidence that our transcriptomic data was a true biological representation of the embryo's status. Here is a little special is chromosome 19, which has the highest gene density, was more inclined to be affected by ploidy variations, regardless of monosomic or trisomic. Next, we asked, are all embryos the same? When we compared day 5 and day 6 euploid blastoses, we found a significant difference with nearly 200 different expressed genes. Functional analysis revealed these genes were primarily involved in critical pathway for placental development, like the PPAR and notch signaling pathways. This suggests that even among neuploid impulse, developmental timing is associated with distinct molecular signatures. When we went a step further, mapping the interaction between the impulse gene expression and the genes known to be critical for endometrium receptivity. On this network plot, you can see the impulse genes, like the color green, talking to the endometrium's green genes in the color red. We identified 3 major interaction clusters, all pointing towards coordinated differentiation. Essentially, as the embryo prepares for post-implantation development, the endometrium is preparing to receive it. This provides a beautiful molecular snapshot of the embryo-endometrium dialogue. Now, for the ultimate goal, could we predict clinical success? We use the data from 83 transferred neuploid embryos to train and test survival machine learning models. We identified 280 genes that were differently expressed between embryos that implanted and those that did not. The results were excellent. The random forest model proved to be the most powerful, achieving an area under the curve of the 0.888 in the test site. This indicates a very strong predictive capability. So, can we use this in the clinic? This is very important. We use the model to create a simple 3-tiered ranking system based on the predicted probability of implantation, good, and the moderate and poor. The results were striking. The clinical prognosis rate in the good group were 88.6% compared to just 50% in the moderate group and only 20.8% in the poor group. This ranking significant outperformed the original selection, which had an overall success rate of 58%. It provides a clear data-driven priority for transfer. In conclusion, our work demonstrates that transcriptomic analysis offers a powerful new layer of information for impulse selection. By integrating RNA data, we can move beyond a simple neuploid or aneuploid determination to a more nuanced ranking of embryos' true biological potential. This has a potential to reduce the time, cost, and the emotion burden for our patients. Is very important for our work. Of course, further validation is necessary. We are now moving toward with larger multi-center prospective studies to confirm the clinical efficacy of this model. Here is our research group, and thank you very much for your time and attention. Thank you.
Thank you for your presentation. Are there any questions? Maybe I'll ask one. So you did biopsies on blastocysts, and you split the biopsy, and with one half of the biopsy you did standard PGT, looked at aneuploidies, and the other half you used for RNA-seq.
Yes, exactly.
But you didn't present the correlation between aneuploidy and RNA results. Did you also see some a correlation like the previously speaker?
Yes, exactly. We after the biopsy, we split samples to two parts. One part for the DNA, like the normal PGT, and another like...
Try to speak
RNA-seq.
close to the microphone.
Yeah. Another is like the, which is RNA-seq to compare the two data. And
then you did find a correlation. You split the group, you analyze the data, RNA-seq, and then you compared it with developing, implanting embryos or euploid embryos.
Sorry, can you
You compare the RNA results between which group? Between the aneuploid embryos or the one that didn't result in pregnancies?
Or maybe we can talk later after the conference. Our boss is A65. We can talk the question.
Okay. Well, if there are no questions, we're going to close this session. Want to conclude?
I want to thank you very much for being here, and thanks to all the speakers for the very nice presentations. Have a nice evening.
Thank you.